Well, that changes everything. Hey, shh. Be honey. Be quiet, honey.
30, a paragraph about this experience would be excellent extra credit.
It's true, and actually that's true for the other class, right? Yep. You guys know. I think there are more.
Sometime either today or tomorrow, I'm going to send out in the PCS majors list the schedule grid for classroom proposed for the fall. If you see any conflicts, let us know. We may not be able to work things out, but it would be easier to work things out now than if you wait till the fall.
Thanks. Yes.
Two sustainability-related events next week. Monday, we have Perspective Series talk. It's about the intersections of healthcare and climate change. And then Tuesday night, we have our first sustainability film of the screening, Evolution Earth, in 106 Zabriskie Popcorn.
Ooh! I heard evolution. I got excited.
Your sustainability talk is Zoom, correct?
Yes.
Okay,
Yes,
so
this one is via Zoom.
there's a Zoom link on there, so why don't we, after this talk's over, we can put that up on the Science Colloquium board out there in case students are interested.
Sure.
They can go and get that, and then we'll pull it down Monday. We'll pull it down Monday after the talk's over.
Oh, I know they are.
So if you guys are interested in the talk, the information will be on the normal Science Colloquium little placard we have out there. So be sure to check it out.
We have another one.
Yes.
Our pre-vet polo meeting is going to be next Saturday. I got your name down if you came to the other meeting on Science Colloquium last week, 2 weeks ago, sometime like that. If there's anyone else interested, just let me know, and I'm going to be sending out an email with the time for it.
Any other announcements before we get started? No? Okay. So today with us, we have our very own Professor Jackie Schnurr. She's going to be talking to us today about the effects of small mammals.
No, that was my dissertation.
Oh, yeah.
Look at the screen above you. Come on, man.
So,
You were savvy about
she went
Schnurr.
to Cornell University. It looks like you BS'd your way through natural resources?
I did BS my way through natural resources.
Oh, I see. She has a BS. Fantastic. So she did that in 3.5 years.
I did.
Very, very cool.
Oh.
After that, she decided to go to a different area, Idaho. So if you guys ever want to know about Idaho and all the potatoes scattered all over there.
That's all I talked about.
So she did. She got her PhD in ecology. She also has a bonus PhD. Let me know what
Come on.
bonus PhD is in.
What do you think, guys? What do you think? Nope, geology's a good guess. Mathematics!
How many degrees do they give out in mathematics?
Oh, I'm the only PhD from mathematics from Idaho State. It's a long story. But my minor was in geology.
2 PhDs.
I'm a paradox.
She chose Idaho because of its proximity to the mountains. She likes to ski, so she did that in her free time, which there's not much of when you do a PhD, but she managed to ski in there. Postdoc following her PhD was at Savannah River
Ecology lab.
Ecology Lab, that's right, in South Carolina. Anybody here ever been to South Carolina? Yeah, it's beautiful.
I feel similar to that as Cornell.
Yeah. It was hot in South Carolina, so she decided she needed a change of pace, and at that point she decided to go to Alaska. She worked in Alaska at Sheldon Jackson College. We'll see a couple pictures of that up on the screen today. She'll be talking about that a little bit. And then after that, she decided to come back to New York, and she is now working at Wells. She's been here since 2004, which means next year her career is old enough to get a beer.
Oh my God. You know, the other night I couldn't sleep and I woke up and I thought, I started graduate school 30 years ago. I just thought I'd say that.
That's okay. All right. So without further ado, we're going to turn it over to Professor Schnurr. Let's give her a nice welcome.
Thank you. Oh, thank you. Thank you, Ben Senior. Well, thanks everybody for coming. This is the event to end the I Heart Wells week, and so I do have some Wells College swag to give out today. So you don't know who's gonna get it. They gave me some raffle tickets, and I was like, I'm not I'm not dealing with that. But anyway, I do have this lovely hat that Ben did not put on his head, a water bottle, some pens, a little bag, cup. So ask some good questions and we'll see who gets to have the swag. So basically, I haven't given you guys a talk in a long time, and I wasn't sure that anybody knew exactly what I did. I mean, I do run around the building like a crazy person most of the time, kind of dance a little bit, but I thought maybe you should hear about maybe what I do for research. So you will realize, and those of you who have had me in class before know that most of my projects have to be outside. I like to be outside at all times. And so most of what I'm going to tell you about is the stuff that I collected in the field. And I've had students do the work in the lab so that I could be outside. So this actually has nothing to do with plant ecology. I put this in for Paul because it was water, but I did take this photo. This is up at my cabin in Indian Lake, and I thought that you guys would think it was super cool. So this is Indian Lake in the Adirondacks. Currently, I don't have any research up there, but that doesn't mean that I won't soon. So the first project I'm going to talk to you about is something that I did starting when I was in Alaska, and I was in Alaska from 2002 until 2004. I taught at a very, you think you guys are small? A very small Alaska Native serving college called Sheldon Jackson College. I'll show you a map in a minute where it was. They had 117 students when I was there and about 15 faculty. So it was not a very large college, but it was extremely fun for my first job. When I was there, I really got interested. Oh, here's Sheldon Jackson College. See, I told everybody earlier that I keep changing my slides, so it's going to be like a fun adventure for me too. Like, what did I put in there? So this is actually the science building. This was the SAGE science building on Sheldon Jackson College's campus. There's the campus back here. We were right on the water. This is the Pacific Ocean. This is a salmon hatchery. So when you asked me about fish hatcheries the other day, I was like, yeah, I know where one is, but it's kind of hard to get to. So this is where I worked. This was my office window. Again, if you turned around and looked out, here's the Pacific Ocean. So this is really the view from there, looking back out towards where the islands were. And we could talk a lot more about how much fun it was to work at Sheldon Jackson College. It was an adventure. But what I really got interested in were these things that are bogs that are called muskegs. And muskegs are these swampy areas that are found throughout southeast Alaska, usually wet, very difficult to walk through, usually full of grizzly bears. So it was kind of one of those things that, you had to be a little careful working in the field. But if you brought a big group of students out, we made enough noise that we didn't often encounter bears. We did encounter like steaming piles of bear sign, but not the bear itself. This, believe it or not, is a type of tree called shore pine. Shore pine is called shore pine in Alaska, but it's called lodgepole pine everywhere else. And so you may be familiar with lodgepole pine if you've ever been to Yellowstone, really big, tall trees. That's not what they look like in Alaska. They look like this. They grow in these really wet, swampy areas, and they grow extremely slowly. So when I was in Alaska, I decided to see how slowly they grew. So I had some students sample shore pine growth, and basically we cut down a bunch of little seedlings, and we saw how old they were. So that was in 2004. In 2019, I thought, I have some really cool data from 2004. It would be really neat to go back and see what's gone on. So I planned this trip, went back out to Alaska, and we ended up doing some more work on the shore pines. So just in case you weren't familiar, by the way, my ArcGIS kids, I made this map in ArcGIS. So this is Alaska, and so where it is, Sitka's down here. Juneau is right there. As a fun aside, imagine going to college there. It's on an island. The only way you can get there is either by ferry or by airplane or boat. You can't drive there. So I had a bunch of seniors at the time who wanted to take the GREs. And they were like, oh, we need to take the GREs. And they were like, okay, just drive over to Juneau. It's only 90 miles away. And my poor kids were like, what do you mean? So they literally ended up having to get a plane ticket to fly over to Juneau so that they could take their GREs over in Juneau. So here's Sitka. This is where I worked. This is where Sheldon Jackson College is. When I did my sampling, I did some sampling in 2019 here in Sitka, but I also did some sampling over in Juneau and also up at the Glacier Bay National Park. So if you are interested in going to the Glacier Bay National Park, you should do it soon. There are tidewater glaciers. Tidewater glaciers are glaciers that go down into the water and float, but they are actually melting so quickly that they probably won't be around for very much longer. So I was like, no, I'm gonna go see these tidewater glaciers before they're gone. So I also went to Glacier Bay National Park and sampled up in Gustavus. This is where actually the home base for the park is, and then you take boats into the Glacier Bay National Park. So when I went back to the data from 2004, it turns out that it wasn't very good data. So I'm not going to tell you anything more about my 2004 data, just that it existed, and that's where the idea came from. So what we asked then is first, is the climate actually changing, right? I had an interesting conversation the other day in my How Do You Know class about climate change. It was fantastic. I loved it. But is the climate actually changing? And so what we were looking at is temperature and precipitation. You ecology kids know that all I do is talk about temperature and precipitation. It kind of gets old, but that's what I worked on, was temperature and precipitation. And then if that is changing, can we see that in trees? And the way that we are looking in trees is looking at tree cores. And so I'll tell you more about tree cores. I'll show you a picture about tree cores. But basically you take a drill and you drill into the tree and you pull out all the growth rings and you end up with a sample that you can then look at and see what's going on with growth. So just so you wanted to see some more pictures, I told you it was my travel log. So this is Glacier Bay National Park. This is actually the boat that you take in to go check out the glaciers. Notice this one's no longer tidewater. There really are only two that are still tidewater. So yeah. This is Sitka. So this is the airport. If you can see this line going into the Pacific Ocean. It's quite an adventure to land when you go to Sitka, because you are, you've been on a plane, you're flying in and you're like, there's nothing under us, guys, nothing. And all of a sudden you're like, whoo, you hit land. It's crazy. Sitka is about 6,000 people. It's the 6th largest city in Alaska. This is up on Harbor Mountain. So this is a mountain that looks above Sitka. And just as a fun note, it was really interesting to do labs there, because I could actually start at the intertidal zone and sample up through all of the biomes up until to the tundra areas. So it was pretty awesome. And then this is Juneau. You may not know much about Juneau. What you can tell, though, is Juneau, it's definitely cruise ship capital of the world. It is the capital city of Alaska, and really it is landlocked. If you haven't noticed that all these places are very much close up to the water, really hard to get anywhere, because if you try to go up, and why this picture looks from like this is that we took a float plane tour above the Mendenhall Glacial Field. And again, I could have given you a whole bunch more pictures, but I was like, control yourself, control yourself. And so just so you get an idea of what these areas look like, right? So there's a whole bunch of different types of trees. These trees, right, are gonna be mostly, and what I'm gonna talk about, coniferous trees. We're gonna talk about Sitka spruce, we're gonna talk about western hemlock, and I'll show you again in a second, and then the shore pine. This is the Tongass National Forest. So you may have heard about logging in the Tongass National Forest. This whole area is the Tongass National Forest. And so logging is important. So again, this is just another view of muskeg with some other of these shore pines, right? They're small. They can be tree type sized. So I had to sample trees that I could actually take tree cores of, which is kind of why I couldn't compare it to the 2004 data, because those were tiny little ones. I didn't have a tree core back then. Sadly, Wells has more money than Sheldon Jackson did. It's hard to believe. And then the other types of trees, right? So this is a Sitka spruce. Super cool, right? Looking up into the canopy. These things are huge. Like, these are like the big old growth trees that you hear about. So they're super awesome. And then this is western hemlocks. I also tried to sample these things called mountain hemlocks. And here's my son actually climbing around on a tree up here with some other kids. The problem with these trees is that because they're up above snow, like in the area, it was near impossible to take tree cores. It was like, Gah! And then when I did get them and I brought them back, they were like destroyed. Like you couldn't even read any of the rings. So unfortunately, the mountain hemlocks didn't work, but it was kind of fun to do. I mean, what do I do? So here's what my tree core looked like. So it's basically just a little tube that's got a drill bit in it. You drill it into the tree, and then you stick that metal rod in, turn it, and you can pull out that tree core. And so you can see even on this, like there are some growth rings in here that you can see and you can measure. And then when I brought it back, I worked with Caitlin Lawrence. Some of you may know her as Larry. And Caitlin Lawrence really did sit in my lab and count every one, measure every one of these growth rings. So she devised her own way of doing it. So this is her microscope. She's got a ruler. She's trying to keep track of those tree rings. She actually was reading it off into her phone and then recording the data in her phone and then writing it down. If this is something that you're interested in doing, since this time, I ended up purchasing a scanner, believe it or not, that you could actually put the tree cores in and it scans it, and then you just have to look at the image and it measures it for you. I know, right? Don't tell Larry. I saw Larry over winter break. She came back with her boyfriend, Derek Shea, and fiance now, but she's almost done with her master's degree out west, so she's really has moved on. And we also presented this work at the Ecological Society of America meetings in 2019, but the meetings were actually in 2020, and something happened in 2020, so it was all virtual, sadly. All right, so data slides, you guys know I had to at least put a little bit of data slides in my photo talk. And you also knew that they had to have some graphs. So anybody who knows me knows I love graphs. If I'm ever going to explain a graph to you, I will make sure to tell you what the axes are. I hate when people stand up here and say, as you can see, because I don't know what I'm supposed to be looking at. So this is a graph of precipitation. So really just rainfall. Similar to the aurora bubble, Sitka doesn't get a lot of snow. It's mostly rain. And then this is temperature from 1949 to 2019, which is when we did the study. And so from 1950, so these are the years, and then on this side is the mean precipitation that was measured at the Juneau airport. You can see that there's a lot of variability in the precipitation through time, but that it is trending upwards. And so it's really increasing by about 0.7 centimeters a year. Doesn't sound like that much, but through time it's going to continue to compound and will increase. Similarly, the graph over here is very similar in that it's the year and the temperature this time. And then this is increasing through time, again, variable, right? You're going to see some variability, but the overall trend is that temperature is increasing through time. And, so now we know that the answer to question 1 was, yeah, it is, right? So what does that mean to the trees? And so you're going to see something similar in the trees. This is, again, year, and this is the tree ring width. So let's talk through the tree ring width. If a tree is growing well, the width would be bigger, right? It would grow more. So the ring, and if the tree is growing poorly, the ring is going to be smaller. So trees that are growing really well are kind of up here. Trees that are growing very poorly are down here. And through time, what we're seeing is that shore pines are beginning to grow a lot less quickly, a lot slower. They're being impacted by the climate. Now, right, I don't know what about it. And what is causing this is a really good question. There's been a lot more studies on this. One of them is about nighttime temperatures, that plants grow usually during the day, but what if it's warm at night? What about winter temperatures? So there's a lot of variability in this data, but overall for you guys today in my photo journal, shore pine are growing a lot worse. What is that going to mean? I'm not sure. I'm sure that probably all of you have heard that Alaska is having more effects of climate change than any place else because they really are warming more than any place else. What is that going to do to the Tongass National Forest? What's that going to do to the whole system with water? I don't know. Take Paul's class. Paul will tell you. He's got all the answers. just give you the questions. Okay, so short pine overall had the same pattern no matter where you were, if you were in Juneau, Gustavus, or in Sitka. The other tree species didn't. And so we broke them out into their different species. Same kind of idea, right? Here's the year, here's the tree ring width, and then this is for Sitka spruce. For Juneau is in red. Sitka is in this tan or whatever, gray color. I don't even know colors. And Gustavus is in the green color down there. And so what we can see is it's not consistent, right? Those three lines are not all going the same direction. They're not actually going on the same slope. They're not doing anything that you would imagine that you saw nicely with the Sitka spruce, with the short pine, right? So in Juneau and in Sitka, they are growing slower. Juneau, it's not significant. So if you're not familiar with regression lines, and I'm sure that most of you are not, right, this is just telling you what the slope of the line is, and this is telling you how well it fits the data. This just means non-significant, but it's still negative. This is a negative number, it's going down. In Sitka, again, we've got a negative number. It actually is a significant decline. Does it have something to do with location? Maybe because Sitka's out closer to the water than Juneau? I don't know. Again, I'm not giving you answers. I'm only giving you questions today, but I'm just telling you that things are happening. But then Gustavus, what's up, right? So here, Gustavus, it's marginally significant. Those of you guys who remember, right, P of 0.05 really is our cutoff. It literally was 0.05. So marginally significant, but it's positive. So what's going on? Why are these trees growing better? I'm not giving you answers, I'm only giving you questions. But what you can see is that something is changing. What's changing even weirder is western hemlock, okay? Same year, tree width, Juneau in the red color, Gustavus. I actually didn't have any western hemlock samples from Sitka, partially because I didn't think to do them when I was there. And when I got to Juneau, I was like, why am I not doing westerns? And it was not necessarily easy to go back and get them. So that's why we don't have western hemlock from Sitka. We did the mountain hemlock in Sitka and it didn't work. But in Juneau, here's our negative slope, significantly growing worse through time. In Gustavus, positive slope, significantly growing better through time. So what does that mean? Yes?
So make sure I understand what I'm seeing.
Yeah.
These are data from, say, one core?
All the cores put together for the different years, yeah. Yep.
It's not like one person did 1950s.
No, it's, yeah, so you can take those cores and you can count back and know what year you're looking at. No, that's a really good question. Sorry, I didn't make that clear. Plant people just sometimes, yeah.
But we've got a couple of questions about the previous graph, because the Gustavus looks like it ought to have a really good R squared, but
Yeah, I don't know why
really
it doesn't.
low. Why it's not up? It's not.
I'm suspecting because it's on the same line as the others. I think it actually has to do with the spread of the data itself. Do you know what I mean? Like, I don't know the answer.
Yeah. And the other thing I was going to say is
Juneau's young.
Juneau, you don't have any old data.
Yeah, those trees were really young.
If you'd had older data, then it might have had more of
Right.
a chance.
So, yeah, 100%.
What was the reason for not, just because it's all, you just happen not to pick very big trees?
I picked big trees, but they just happened to be growing quickly. Perhaps it has something to do with the glacier movement. I don't know the answer why.
Right.
But I was sampling near. I had to sample in areas where I wouldn't get caught. Because I didn't have permission. But you're right. I mean, there are so many more things that we could go back and do.
Yeah.
I just haven't been able to get back and do it. Ben Jr. wants me to go do that, but. Yeah, no, he's all about. In my defense, I have a friend who works for the National Forest Service, and I said, hey, do I need a permit for this? And he was like, not if you don't get caught. So, so in my defense.
I have a friend who's a lawyer.
I have a friend that's a lawyer. Come on, Lex. All right, so yeah, so I mean, there's obviously a lot of stuff that's going on here that could be looked at more. I don't know the answer. I wish I did know the answer. And honestly, right after I did this, I was like, I'm going back. And then this weird pandemic happened, and then everything kind of like went on the back burner. So it hasn't gone away. This kind of a study is something that you could do at any time because those trees are still there. So you could go back and look for some holes, see which ones I sampled.
Did you find the same trees, or did you label them anyway?
No, if I labeled them anyway, they'd catch me. Yes, Chris. Yeah, you're right. That's a good question. Let's go. There was like a weird little blip. A weird little blip in precipitation, but not in temperature. It's a good question. Some sort of threshold was crossed. Interesting. I wonder if we broke it there, if we could see a different pattern. Look, relatively flat and then increasing. But yeah, it's a good question. We hadn't looked into it that way, but. Other questions? Because this is actually my last Alaska slide.
I'm just curious if you're seeing any hemlock woolly algae infestations in those hemlocks.
That's a really good question, and it's a really interesting question because the guy that was supposed to be giving this talk, who canceled on me 3 times, Mark Whitmore, is actually there. Because the western hemlocks have a bug called Larobus that controls hemlock woolly adelgid. So what he does is he goes over there and he collects the Larobus and brings them back here to try to help our hemlock. So that's actually why he's not here. So no, they don't have that problem because they have a biological predator. Yeah, no, that is really interesting. There is something happening to the hemlocks though, and I didn't talk about that, but in Gustavus, the hemlocks look terrible. And I was like, what's wrong with these? And I went in and I talked to the forest ranger and he was like, we don't know. And I was like, all right, cool. That's cool. So yeah, so this is just like, there are more questions. What's Alaska going to look like, So this is one of the prayer houses. So Alaska Native culture is extremely important in this area. This is Tlingit. And so you'll see the totem poles and they have the symbols. And so that's why I have my Alaska Native bracelet. I also have a very large tattoo on my back about that. But aside from that, I just wanted to say that, there's a lot of cultural things that are going to change as well as biological things with climate change. And that's just yet another thing that we need to look at, is putting the science and the culture together. So anyway, it was fun too. It was super fun. You should go. Shifting gears. So this is, you all know and love, pale swallowwort. It's lovely, right? Yeah, it's really pleasant. This actually is from behind McGordon House. This is really from right here. So all of these species right here, not the yellow flowers, this is pale swallowwort. Invasive species, actually came over to us because it's pretty, it has pretty flowers. People planted it in their gardens. It has escaped. Big problems with it is it's in the milkweed family. And so sometimes monarchs think that it's a cool thing to lay their eggs on. It doesn't have the same kind of properties as milkweed does, so helping with the monarch butterfly decline. Also, and here's where things get a little weird, people always think that this is bad and that it's taking all of the native species out. Really great question. That's actually what my data here is all about. Perhaps some of you have seen the swallowwort plots, right? If you're walking down Woods Path, off to the left, there are some weird pipes in the ground and some little containers. Yeah, those are mine. We also have some over at McGordon House. We have sites at Long Point, Owasco Bluffs, Green Lake State Park, Clark Reservation State Park, along the sides of roads, places. We have sites all over the place. This is a group that's working on biocontrol. We also had these cool tents set up for a while. We released these moths inside those tents, and they did a really good job, the caterpillars, not the moths, did a really good job eating the swallowwort, and then they disappeared. And so this is a project that we started in 2018. It's me, Andrea Davalos at SUNY Cortland, Dylan Perry at ESF, Carrie Lima Brown at Cornell. Which disappeared? The swallowwort, I wish, disappeared. The moth. Yeah. And then so there's one more. Lindsay Milbreth down at Cornell, USDA. agricultural research station. But, so we released this biocontrol agent here on campus. This tent was set up. Story, they mowed my plots. Several times they mowed my plots. And who was it, Gary? Who does the mowing? You guys know what I'm talking about? Anyway, yeah, he was like, it's okay, I didn't hit the tent. And I was like, well, why would you have hit that tent? Like, why would you have mowed that? Why the hell did you mow? Anyway. And actually, even worse than that, completely off topic, we had set up a really great site over at the plots by the the farmhouse, whatever you guys call it over there. They were awesome. It was great to work. And then they mowed the whole area. And so I had called Brian Brown and I said, what the heck, why'd you mow it? And he said, oh, I'm sorry, you want me to plant more? And I was like, no! I was like, huh. So this project has been ongoing. We did have a $750,000 grant to do work. My part of the project is actually looking at native plant regeneration after the biocontrol agent worked. The biocontrol agent didn't work. So there's really nothing out there to change. These are two of my environmental students, that's Cheyenne Nowalk and Gabby Sawyer. That's a point quarter frame. They spent a summer out measuring all of our plots, trying to figure out what was going on with the native plants. They did the first data. I did the second data this past summer, and there's nothing going on because, well, the biocontrol didn't work. The project is actually in iteration part 2. Now we're bringing in a beetle to see if the beetle will do it. But if you know anything about biocontrol, it's not a fast process. Right now, the biocontrol is actually at Cabby. Cabby is in France. They do a whole bunch of work. They bring in our native species there. They release everything. They see what the biocontrol agent might possibly eat. Then they can bring it into our area. A couple of places are quarantined. Cornell has one. SUNY ESF has one now. Then they'll test them here. And then after a long time, eventually we're allowed to release them. So part 2 of the project probably won't start for a couple years. But that's not what I was here to tell you about. There is the idea that these invasive species are bad, right? They're killing our native species. They're really bad. So one of the guys that I worked with, Lindsay Milbraith at Cornell, in 2009 started looking at this. And if you work for the USDA, sometimes they decide all of a sudden that you don't work on that anymore. So he was working on swallowwort, and they were like, no, now you work on this. So he was like, well, what do I do with all this data? So he gave it to me. So I have a whole bunch of data from the Musgrave Research Farm. Some of you have been there. It's over on, thanks, Brad, on Poplar Ridge Road, right? It's the Cornell Research Farm. That's Musgrave Research Farm. And then the other site we have is Waylay State Park, which is up in the not really the Thousand Islands, kind of the beginning of the Thousand Islands, right on Lake Ontario. And so we have 2 projects there looking through time. I'm actually working with Burwell's husband on this. He's helping me a lot with data analysis because he's a statistician. So basically, I'm asking, does pale swallowwort affect spring ephemerals? I was wondering if you guys can read that font. I was like, oh, it's cursive. So does invasive pale swallowwort affect the abundance of spring ephemerals? I'll tell you what those are in a minute. At those 2 sites. And then, basically the same kind of idea. If there's no pale swallowwort, do you have more of these spring ephemerals, which are just plants? And then part 2, what about during the summer? Are they really affecting plant diversity through time? So those are the 2 main questions that I'm answering with this data. So spring ephemerals are pretty much my favorite. They're my favorite. So as soon as it gets to be warm, these guys come up. They're awesome. They grow, they flower, they set seed, and then they die before the leaves even come out on the trees. Super cool, super fast, right at the end of the semester where we're all working really hard, so it's really hard to go study. But they're awesome. Anybody who's gone for a walk up to the waterfall, remember that, you guys? Remember we saw all the spring ephemerals, right? So there's a lot of spring ephemerals up there. This is bloodroot. Love them. They have these little leaves that This is a hepatica, sharp-lobed. This is, believe it or not, wild oats. So you really can sow your wild oats around here. And there's a cute little swallowwort growing next to it. And then these are Trillium. You're probably all familiar with Trillium tree. Try 3. They have 3 leaves. They have 3 flower petals. I just like the word 3. So this is the data from Waylay State Park. These are the different species on the x-axis, and this is how many plants are in each of those areas. From 2011, 2015, 2017. 2009, they sampled weird. And so I pulled that data out. It's not that it didn't show the same kind of pattern. I have no idea. It's just the way they sampled wasn't the same, and I was like, I'm not going to compare that. And then these are different species. This is spring beauty. This is trout lily. It takes me a while to translate into English. This is the sharp-lobed hepatica. Wild oats. Cardamine is cutleaf toothwort, if you know what that is. Dicentra is a bitter cress, and thalictrum is blue cohosh, but there's not a lot of it. Waylay State Park, we don't have any controls. It's all invaded by pale swallowwort. And so what you're seeing is that it doesn't matter, right? Nothing's changing. If anything, they're getting more through time. So the idea here is, so these are the 3 different years. Spring beauty populations are growing up. Trout lily populations are growing up. They're not doing anything different. They're growing better. I think it has to do with weather more than anything else. Yeah.
Do you have counts from the same plots on amount of swallowwort? Is that also unchanging over?
So the swallowwort's not up yet.
Oh, sorry.
Yeah, so that's something that's interesting about the spring ephemerals. The swallowwort's not there yet. So you wouldn't expect it to have any effect. But I will say, because there's no controls here, it makes it a little harder. I told Ben that I put this graph in because I hate when people put graphs in like this, because you're like, you can't see a damn thing on that graph, right? And I know that, and I don't care. Really what I want you to do is to just look at bars. So this is different species, right? Now we have swallowwort and control, If anything, there's no difference at all, right? It doesn't matter. Swallowwort's not really impacting it. Sometimes the swallowwort has more. Sometimes the control has more. But then the next year it changes. So really with spring ephemerals, there's no real pattern here. And that's all I wanted you to get out of this graph. Plus I thought it was fun to put a graph in that I hate. So there. So what about during the summer? During the summer, this is what it looks like out there. It's pleasant. It's really fun to walk through. Trips you. My ecology kids know because we measured it. Remember that? Remember how fun that was? And so invasion by pale swallowwort did not have consistent effects. This is actually looking at diversity. I'm not going to get into diversity indices with you guys today, but diversity indices actually takes into account how many of a plant there are and the proportion there are in the environment. Lots of different diversity indices. I'm going to be using the Simpsons index just because of the way that the data were distributed. Again, not going to tell you more about that. This is the forested plot. This is Waylay. And this is Great Gully. You guys know where Great Gully is, right? It's right here, right up the road. So Musgrave is in blue. That's the forest. Musgrave. Cornell has a control. Remember I said that. And then this is the Waylay plots in gray. And really the idea here is that their swallowwort is not having any impact. So if you compare the blue to the red, the blue to the red, the blue to the red, the controls have less diversity than the plots that have been invaded by swallowwort. It's not what anyone expected. In the field, so what's going on is, at Waylay at least, there is what you expected. So plots that have a lot of pale swallowwort actually have decreased diversity. Hey, that's kind of what we all thought was going to happen with invasive species. What the heck is going on? And then if you look at Great Gully, again, it's not what you would imagined. At Great Gully, it's the same kind of idea. So why does Waylay show this pattern that we expect and Musgrave and Great Gully, so here, doesn't? I don't know. But I have a guess. I actually think it has to do, and I was complaining about this map, with soil. I don't know if you guys know anything about soil. If you go out there to try to dig a hole, you can dig for a while, usually before you hit ground, right? You can be out there. You can push those pipes into the ground for quite some time before you hit dirt or rock. At Waylay, the rock is on the surface. There is absolutely no soil. And so what I think is that environmental conditions are actually driving this pattern. That's where I'm at with this paper right now. I really am working on this paper. I think the title of my poster on this was "Invasion is not a monolith." In other words, there's not a right answer at all times. I think that it's going to matter based on where you are in the environment. Questions on this one? Okay, yeah? July, so growth in the middle of the growing season. Yep. Good question. Yeah, the ephemerals are gone. They'll be gone by the end of April. All right, so the final project, and one that I think is super cool and happening right now, right this minute, as we're doing. So this is the Mohonk Mountain House. Some of you may already be familiar with the Mohonk Mountain House. The Mohonk Mountain House is this amazing resort found outside of New Paltz. Lots of fun, cool things about this place. It's owned by the Smiley family. Literally a hotel that it's owned by a family. They have their own quarters. They come in. The Smileys are there all the time. They greet guests. I know them. They know me. Like, it's a really homey little place. You pay a whole hell of a lot of money. I mean, like over $1,000 a night to stay there. That comes with food. Not alcohol. But food. But you can use their grounds. So you can do whatever you want there. You can hike. Any rock climbers in here? So a via ferrata, I don't know if you know what a via ferrata is. It's terrifying to me because I hate rock climbing. But they have like these ropes and stuff along the side of the cliff that you can climb on. I'm going to show you some cliffs in a minute. But they're the only place, at least in the East Coast, that have a via ferrata. And you can do these things as hotel guests. Why do I go here? Yeah, because I'm rich. Sorry. So they hire me. I lead nature walks for the guests. So if, again, any of you know me, I love to be outside and I love to tell everybody about everything. So I lead walks around this and talk to people, and I don't stop talking. And they ask me questions, and I have a really good time. My last sabbatical in 2018, I thought, wow, wouldn't it be cool to write a field guide to the Mohonk Mountain House grounds? And so I did do that, and then that pandemic hit, and whatever. But as part of that field guide, I learned about a species called mountain spleenwort. And mountain spleenwort is a fern, and it is endemic to the New Paltz region. Now, what does endemic mean? Only found. This is an iNaturalist map. Those of you who know me know I love iNaturalist. You can search iNaturalist for species. So what I did was say, explore. Tell me where mountain spleenwort is found. So this is us over here. Right? This is New Paltz down here. In New York State, mountain spleenwort is only found in that region of the state. So because of that, they considered it threatened. They think it's a threatened species in New York State. It's imperiled. It's something that they just don't know a lot about. So when I started going to Mohonk, I was like, man, it would be super cool if I could find this really cool little species that we find around there. I got to go through this quick, man. You guys made me talk a lot. And so this is what mountain spleenwort looks like on the cracks in the rocks. They form in the cracks of the rocks, tiny little fern. And by what I mean, oh, and this is Mohonk. This is like, there's one of those walkways around the cliff. I mean, this place is amazing. It's like, anyway. This is also for geology kids. This is a conglomerate. It's something special about it. It is silicate. What does that mean? Our rocks around here are all limestone. So this plant can't grow around here. It can only grow in very specific places in silicate rocks. So anyway, here's mountain spleenwort growing along in these cracks in the rocks. This is how big it is, right? Look how super cute it is. Look at all those cute little sori on the bottom of the leaves. It really is a fern. It's super awesome. Anyway, so what I want to get to something cool. What I'm trying to get to is trying to figure out if mountain spleenwort is stable. Right? When I first started looking at mountain spleenwort, I knew where one plant was. I now know where hundreds of these plants are, but how many more are out there? And I just need to know. Why do I need to know? This area is really popular for rock climbers. I mentioned that before. This is the Mohonk Preserve. This is a whole line of mountain rock climbers just trying to climb up these rocks. This is a crack in the rock. This is all chalk. Hey, where does mountain spleenwort grow? Oh, look, in the cracks in the rocks. And so what we think is that the more rock climbing happens, the less populations of mountain spleenwort we're going to have in these areas. So, and again, if you go to Mohonk Preserve, not the mountain house, that's different, you can get these rock climbing routes. We know where the routes are. I just took that picture because I like to. And this is my friend Liam. Liam is helping me sample these things because I kind of mentioned already that I don't like rock climbing. And I don't really want to climb up those things. And so what am I doing instead? Come on. Make it bigger. So we're using drones. And so Liam is my drone operator. He has his FAA license. And he goes out and he takes my drone and he flies it off into the distance. And so what do you see from up there then? You guys really wanted to know this. I know, this is really what we want. So now what is that drone looking at? So this is the cliff face. We just did this in October. Honestly, there's some rock climbers. Here they are. Right? I'm not doing that. But anyway, so, right. What we learned from our sampling, and again, this is the first time we did it, it was not easy to get the permitting to do this, right? You have to have your FAA license. Aside from that, peregrine falcons like to nest on these cliffs, so you can only sample in times when the peregrines aren't around, so we're pretty much stuck to sampling in the fall. But what I want to do is use these drones to fly in and sample all these cliffs. I mean, look at that cool little tree just growing on there, that cliff. And so what we can do is get closer and closer and closer to the sample. The black stuff is all lichen. That's a whole other fun story. But what I'm trying to do is see how much mountain spleenwort is actually on these cliffs using this drone technology to go in and say, all right, what do we got, right? And even you, in your newly untrained mountain spleenwort, none of this is mountain spleenwort. There's none of this is mountain spleenwort. Is that mountain spleenwort? I don't know. And so what we're trying to do is come up with a way of sampling. Sometimes science things become more obvious after you do them. We went up and we did this. It was super fun. What a beautiful day. Got to fly a drone and sample on these cliffs. Why didn't I go somewhere where I knew there was mountain spleenwort? Why didn't I go somewhere where I was like, oh yeah, that's it? I don't know. I didn't. So that's the next thing, but I can't do it until next year because the peregrines are back. I literally wrote to Megan, who's my contact there, and I was like, hey, are the peregrines back? Here's the Hudson Valley for you. New Paltz is kind of over there. But yeah, so this is really super cool, right? And I don't think anyone has ever used drones to sample plant cliffside vegetation before. I think this is a first. So again, this is a thing that's in the works. Do I have any data from it yet? And again, remember, Liam is back on that. I showed you where he took off from. He's like this is like a video game thing, right? So he's just kind of sampling. Anyway, I'm going to stop my video just because I have one more slide. Yeah?
Do you think they would make it like a protected place so rock climbers can't? No? Okay.
Economically speaking,
Okay.
rock climbing brings in a lot more than mountain spleenwort. So yeah, I mean, I have one more slide for you. Maybe two. I didn't even get my thing. So right, so we're ending up with things like this now, samples where you can go in and I can zoom and I can see what I've got. But again, the next step is really to go to a place where I know there's mountain spleenwort, and I know there's mountain spleenwort, and I have friends that are rock climbers, and they told me where it was. Why didn't I put those together? I don't know. And this is something, actually, I just thought that was a cool picture. This is the lost city at the Mohonk Preserve. So how do you fly a drone in here? You don't. I just climbed up and down the cliffs. That was actually fun. This has nothing to do with mountain spleenwort, but I didn't want to not give a shout out to my cow projects because I do also have these cow projects going on. You know, it's not plant ecology, but kind of. So we have a couple of projects looking at sustainable usage of manure. This was a composting one. This is Pheasant Farms. Pheasant Farms takes their cow poop. Anybody who's been there, Brad left, you can tell what that smells like just by looking at that picture. And they actually take out the solids, and then they put the solids through a digester, and then they take the solids and they make it into bedding for the cows. So it's a completely contained system. We have a second project coming up, actually making biochar from manure. So that one's coming up too. I work with a company called Nutrient. They're USDA, so that's different. And it's I Heart Wells Week. Mic drop. I got mine too. So we have these wonderful things. Whoever asks a question gets a thing. So ask a question. You all asked questions already. No? Yes?
Quick question. Observation. I was hiking Nut Ridge, whatever, and
swallowwort.
I know.
And I thought, oh my God,
It's awful.
it's a death zone, right? But I wasn't there in the spring, so.
Yeah, and honestly, I think here you might not see them, but I think the natives are still there. I think they're like, it's cool, we'll just be friends.
Yeah.
Yeah.
Really dark, shaded and dry.
Yeah. And so it could be an environmentalist too. I mean, and again, I was, I'm trying to think back of where do I see it, and it's those dry areas.
Second thing, I don't need a pencil.
Too bad. Yes. What? Oh, what's your second question?
I'm just wondering about salt air affecting.
Tree growth. My guess is that but they've always grown there.
Yeah, but with temperature.
Yeah, exactly. And that's part of. Yeah, you're right. Good question. What's up, Valley?
This is more of like a general question.
Yeah.
I don't know much about New York species. So, like, how many invasive species are actually in New York? And also, and then also, do you think the beetles will actually work to...
On the hemlocks? Well, Mark will get here one of these days. But yes, that's actually a success story right now, is that the only place they've seen the beetle over winter is here on Wells campus. And they found one over at Fillmore Glen. So it actually even dispersed from here over to Fillmore. So that's super cool. Nothing.
Probably on the back of your car.
My back of my car? Oh, because I go from it. You're right!
You're bringing it around.
What are the odds of these beetles, if they eat enough of these, them becoming an invasive species?
They won't, because they only eat the aphids, and once the aphids are gone. It's a success story, like purple loosestrife, if anybody's familiar with purple loosestrife. So it used to be all over the place. They brought in some beetles. It ate the purple loosestrife. The purple loosestrife dies down. The beetles move on to another area. The purple loosestrife grows back up. Beetles come back in. So it's actually just become basically a predator-prey cycle. I'm coming up here! Oh, can I do light? I don't know the answer. This is the first time that I've been trying to do this. That's a good answer, the question though. Yeah, what's up?
Do we know why the moth died?
We have a suspicion why the moths died. We think the small mammals ate them. But it's a good question. That's actually something that we've been trying to figure out. But honestly, if you want to know the real, my feeling, it's not going to work. So even in a cage where it was just them, they ate it all down, and then the next year the swallowwort grow all back up again. So it's not going to work.
With the glaciers,
Yes.
in those trees,
Yes.
the temperature, did you look at how the cold temperatures affected the ground? And
I did not, but you know what? They make these things called eye buttons that are really small. You could dig them in the dirt, and they would just collect data for me wherever. I got like 15 of them upstairs. Let's go do it. All right, come on. Yeah.
I know you're going on sabbatical
Yeah?
next year. What are you
Gonna do on my sabbatical?
research are you going to do during that?
Probably some of the drone stuff. Maybe I'll have to go to Alaska. I'm going to Italy. That's a good question, Helen. I don't know the answer, really. What I'm supposed to do is figure out how to teach ecology better. I could do that from Alaska. My kid has to go to school.
Oh, it's very limited.
Yeah, you know, this children thing.
This school's in Alaska. For a semester. No children.
All right, who wants the hat and the mug and the stuff?
I want the hat. Which question?
You want the bottle?
I got a question for the
Yeah.
hat.
Oh, and that's tote bag. It's vintage.
Vintage tote bag.
Alum engagement, reunion, tote.
If you guys have to leave, I feel bad you're all still here. Who wanted the mug? I heard someone. Uh-oh, we have a fight. Jendal got it first. Do you want a pen? Wait, where'd the question come from? Yeah.
Out of all of the things that you talked about, which one's your favorite?
It's whatever I'm working on at the time. I really have a problem focusing, if you haven't noticed. I think the drone stuff is super cool, but then when I go back to the Alaska stuff, I was like, man, that was super cool. I like the invasive species one just because I always thought it was bad, and it's interesting to hear that it's not always bad. Cows I kind of like, they smell bad. Yeah, they really do. I'm working with Bailey too. I didn't even talk about Bailey's stuff about her environmental DNA. I mean, I got all sorts of stuff going on. See, you all thought I did nothing. Yeah, Dan?
If you attach a drill to one of those drones, you get 3/4 higher than other things.
I think the problem would be keeping it still. And also the drone itself is pretty light. So I think there might be problems with that, but you know, I'm willing to try.
Why drones? What made you think of drones?
I'm not climbing up there. I actually was thinking, like, if we're trying to sample areas that are inaccessible to most people, like, so my original idea was the rock climbing routes would be different than other routes. And so what I wanted to do was do a comparison. But right now, I'm not even at that point. Right? I got to test the method before I can even get to that. Helen?
I know you said that they wouldn't like protect the mountains, the plants.
Mount Sweetwater, yeah.
Yeah, but because of the peregrine falcons, how does that affect
They close the climbing for the falcons. It's interesting. Like, so that, like, this route is closed on these dates. But for the plants, I think that if I did education, because all the rock climbers that I talked to about it have been really interested more than anything. Like, oh, wow, yeah, I'll keep my eyes open. I'll let you know. So I think that education would be better there.
All right, folks, we are out of this.
I know. Jeez.
We'll give her a round of applause.
Thank you, thank you.
If we have additional questions, she'll stick around for a few minutes. Maybe she can answer.
Yeah, I have to go do a nature walk with the kindergartners in 10 minutes.
Go see the beehive.
Yeah. Free food.
Thanks, everybody. Thanks for coming.
Thank you, great job.
Great, really great.
Well done, oh my God. Okay, all I want to do is come back and do a BS. My God, you guys are so awesome. I'm finishing my master's degree.
I didn't know any knew what I did. This is like a surprise.
Yeah, so
Everyone
awesome.
thought I was really good.
Thank you.
You like this?
Oh, yeah, so awesome. Yeah, I want all of you as my professors, honestly. You're just so awesome.
It's all right. Cut it off.